you can rely on them to take care of necropsy and histopathology analysis and interpretation, including the phenotype characterization of transgenic animals. Assistance is also provided with analysis of clinical pathology data. Other services include gross and microscopic digital imaging, consultation regarding study design, and assistance with grant and manuscript preparation. Staff pathologists can also work as co-investigators on funded research projects and grant applications.
We are interested in the cancer-causing agents or carcinogens present in tobacco products. And one class of carcinogens is called tobacco-specific nitrosamines. These compounds are present only in tobacco products. You don't find them anyplace else. We wanted to use the F344 rat to test the carcinogenic activity of the tobacco-specific nitrosamines, compounds that occur only in tobacco products. And the two compounds that we're most interested in are called NNK and NNN, because in previous studies we had indications that these were actually very strong carcinogens. So we carried out studies in which we evaluated the carcinogenicity of NNK given at only 5 parts per million in the drinking water. So we add the NNK to the drinking water of a group of rats. The rats begin to lose weight, so we know they're getting sick. So at that time, we terminate the experiment, and that's when the histopathology facility comes in. They will examine each tissue of the rat to see whether there's any tumor formation.
These studies were large, complex studies, and we use just about all the services that we've got in the Corps. So for these studies, we had animals that had been in a 2-year, 70-week carcinogenicity studies. So we did necropsies in these animals at the end of the study, which involved gross pathology, looking at all these tissues to see what was abnormal at the time of necropsy. And then collecting and processing all these tissues to slides, to H&E stain slides, which is the standard way you analyze tissues. And then immunohistochemistry. For this study, we developed some new immunohistochemistry markers and employed them and got some very interesting findings with those. And then also we were involved in pathology evaluation, so we had to look at all these slides and find out which tissues had tumors. And so it was really a complex study. What we did was really a specialized service that Dr. Hecht wouldn't have been able to do on his own. It really needed a specialized lab to do it, and highly trained personnel to be able to do what we actually ended up doing.
So this slide shows the lung of a rat that was not exposed to NNK. This was just a control rat that just had normal drinking water. This is the lung of a rat that was exposed to 5 parts per million of NNK in the drinking water. This shows multiple tumors in the lung, and the histopathology facility is absolutely essential to evaluate and characterize these tumors. It's a very specialized skill. they are able to classify them as to what kind of tumor they are. Adenoma, adenoma with atypia, adenocarcinoma, which is a malignant tumor of the lung, squamous cell carcinoma. As it turned out, most of these were adenocarcinoma, which is actually now the most common type of lung tumor that's seen in smokers. We carried out a similar study with NNN in which we administered the compound to these rats over a period of multiple months at a dose of 12 parts per million in the drinking water. And the NNN at this dose turned out to be a very powerful oral cavity carcinogen. It was very specific. And this just shows one of the oral cavity tumors. This was in the tongue, induced by NNN. Again, the histopathology facility is absolutely essential for this work, to be able to histologically characterize the tumors and tell us whether they're benign and malignant and what kind of tumor they actually are.